Welcome back to the deep dive. We've had some really interesting questions coming in lately.
hmm
And one that a lot of you have been asking about, especially after our, initial chat, is how we can understand and maybe support our bodies when it comes to these spike proteins.
Yeah, that's definitely been a hot topic.
And it turns out there's some pretty compelling new info that builds right on this.
Exactly. So for this deep dive, we're going to look at a scientific paper, fairly recent, from the latter half of 2024.
Okay.
And this research really focuses on how we might sort of encourage a natural detox from spike protein. Specifically, it's honing in on the health and recovery of our mitochondria.
Mitochondria,
Right. It's
right.
actually a fascinating follow-up to some of the ideas we saw discussed initially on that Marriagenomics YouTube channel.
hmm
But now it's, backed by more in-depth scientific work.
Okay, so mitochondria, just a quick refresher for everyone
Mhm.
listening, these are basically the tiny engines inside our cells, right? Making energy.
That's the core job, yeah.
Yeah.
Cellular power plants.
And we've talked before about how, well, problems with these mitochondria, this energy production, might be linked to all sorts of health challenges.
Absolutely. There's that growing idea, particularly around cancer being potentially a metabolic disease tied into mitochondrial function.
Right. So the idea that spike protein could be messing with them, that's definitely something we need to get a handle on.
For sure. Now, just to be clear, in this deep dive, we're not really going into the super deep weeds of how spike protein might cause the damage.
Okay, good distinction.
Yeah, that's a whole other complex topic, maybe for another time.
Got it.
Instead, our goal today is really to explore the potential strategies for protecting against this damage, specifically focusing on mitochondria.
So looking at the solutions part.
Exactly. We're basically going to unpack the solutions that are laid out in a key figure in this new paper.
Okay, so this isn't just, here's a potential problem. It's more about what can we potentially do? And this research sounds like it offers some, understandable and maybe even actionable insights, ways to support our own cellular defenses.
That's the aim. The authors talk about something they call spike-associated pathology.
Okay.
And this term covers things we've heard more about, like long COVID, but also this newly recognized term post-vaccination syndrome.
Right, I've seen that mentioned.
What's really useful, I think, is how they've outlined the main ways spike protein might be causing problems, specifically for our mitochondria.
Okay, yeah, let's break that down. What are the main ways this protein could disrupt our little cellular powerhouses?
So the paper points to four key mechanisms based on their figure. First, just direct interference. The spike protein can directly mess with how well the mitochondria actually function.
Okay, direct damage. Makes sense.
Second, it seems to hinder something called mitophagy.
Mitophagy. Sounds like autophagy.
It is. It's a specific type of autophagy that's the cell's general cleanup process. We'll get more into that. Mitophagy is specifically targeting and removing damaged mitochondria.
Ah, okay. So spike protein stops that cleanup for mitochondria.
It seems to inhibit it, yeah. The third issue is that it looks like spike protein can also impede the generation of new healthy mitochondria.
So stopping the replacements from coming online.
Kind of, yeah. And finally, the fourth way involves these aggregates of spike proteins, clumps, basically.
Okay.
These can contribute to forming microclots, which,
Yeah.
we did a whole series on that before, if you remember.
Yeah, the microclot connection definitely rings a bell.
And those microclots can then lead to hypoxia, just, reduced oxygen getting to tissues, which is obviously bad for mitochondria too.
Okay, so direct damage, blocking the cleanup, stopping new ones being made, and these aggregates causing clots and oxygen issues. That's quite a list.
He covers the main concerns related to mitochondria, yeah.
So those are the challenges. Now, the important part, potential solutions. The paper lays out strategies to counter these effects, right? You
Yeah.
said four main ones.
Well, broadly speaking, yeah. The first strategy is really about directly breaking down the spike protein itself, getting rid of it.
Okay.
The other approaches are more about counteracting the damaging effects it has on our cells, particularly the mitochondria.
Right. Let's start with that first one then. Directly degrading the spike protein.
Sounds like a plan.
What does the paper suggest for, actually tackling the protein itself?
It points to two main things here. First is nattokinase.
Nattokinase, heard of it. It comes from that Japanese food, natto. Fermented soybeans.
That's the one. It's an enzyme, specifically a protein-specific enzyme, meaning it breaks down proteins.
Okay.
And lab studies, it's important to note these are lab experiments, suggest it can target and break down spike protein.
Interesting.
And what's also interesting is that nattokinase is known to have thrombolytic or fibrinolytic properties. Basically, it can help dissolve blood clots.
Oh, so it might help with those spike protein aggregates, the microclot issue too?
That's the potential double benefit, yeah.
Yeah.
Targeting the protein and potentially the clots it might contribute to.
Okay, that's significant. What else was mentioned for direct degradation?
Aspirin was also mentioned, potentially for helping clear those aggregates as well.
Okay, so nattokinase and maybe aspirin for dealing with the protein and its clumps.
Got it. Now, what about counteracting the damage it does? You mentioned autophagy earlier.
Yes, autophagy. It's basically your cell's internal recycling and cleanup system, a fundamental process.
Cellular spring cleaning.
That's a great analogy. Yeah. It's how the cell gets rid of damaged parts, misfolded proteins, old organelles, just waste and non-functional stuff.
How does that actually work, like inside the cell?
Well, simplified, when something needs to be removed, it gets tagged or marked. Then a sort of membrane bubble forms around it that's called an autophagosome.
Okay, a little garbage bag.
Kind of. Then this bag fuses with another part of the cell called a lysosome. And the lysosome is full of powerful enzymes that work in an acidic environment to just break down whatever's inside the autophagosome. Wow, so it captures the trash and then dissolves it.
That's the essence of it. And this is
Yeah.
happening constantly, just maintaining cellular health.
And you said there were different types.
Broadly, yeah. There's selective autophagy, where specific things are targeted. Often they get tagged with a little molecule called ubiquitin.
Okay.
Mitophagy, getting rid of damaged mitochondria, is a perfect example of selective autophagy. The damaged mitochondria get tagged, then removed.
Got it. Specific targeting.
Then there's non-selective autophagy. This is more of a bulk cleanup. It often gets ramped up when the cell's under stress, like when nutrients are scarce,
Right.
like during fasting.
Ah, okay. That makes sense. So how do we encourage this autophagy, this cleanup, to maybe help deal with the effects of spike protein?
Well, the paper really leans into lifestyle interventions here,
Yeah.
which is interesting because it puts some control back in our hands,
Right. Things we can actually do.
Exactly. And one of the most powerful ways to induce autophagy is through fasting.
Fasting.
This can take different forms. You get time-restricted eating or TRE. That's where you just limit your eating window each day. Maybe eat only within, say, an 8-hour window or 6 or even 1 hour for some protocols.
Right. Compressing the eating time.
Then there's intermittent fasting, or IF. That usually means longer periods without food, like a full 24-hour fast, maybe 36 hours, or even longer sometimes.
Longer? How long are we talking?
Well, the paper mentions potential benefits from even longer fasts, sometimes up to 14 days in certain therapeutic or supervised contexts. Obviously, that's not something to just jump into.
Wow. Yeah, 14 days is serious. So what's the link? Why does not eating trigger this cellular cleanup?
It mainly comes down to a key sensor pathway in our cells involving something called MTO. That stands for a mammalian target of rapamycin.
MTO. Okay.
Think of MTO as the cell's growth and abundance switch. When there are plenty of nutrients around amino acids, glucose MTO is on.
Right. Signal says grow.
Exactly. Build stuff, grow, divide. And when MTO is on, autophagy is generally turned off. The cell doesn't need to recycle if resources are plentiful.
Okay.
But when nutrient levels drop, like during fasting, MTOR gets switched off.
And that's the trigger.
That's a major trigger, yeah.
Yeah.
MTOR off, autophagy on. It's like the cell saying, okay, resources are scarce, time to tighten our belts, recycle what we can, and get rid of any damaged or unnecessary parts to conserve energy.
That's a really clear explanation. Abundance off, cleanup on. The paper even mentioned cancer in this context, didn't it?
Yeah.
How cancer cells often have MTOR stuck on.
That's right. Many cancer types show overactive MTOR signaling. It helps drive their uncontrolled growth, but it also means they often suppress autophagy.
Which might normally help clear out problems.
Potentially, yeah. It's why things that influence MTOR are such a big area of research, not just for general health, but for cancer too.
Fascinating. Okay, so fasting is a big one for autophagy. What about diet? Specific foods or compounds?
Yeah, the paper lists several things found in food. Spermidine is one. It's found naturally in things like wheat germ, aged cheese, mushrooms, though there's some debate about how effective supplements are versus getting it from food.
Okay, spermidine. What else?
Caffeine. Your morning coffee might be helping.
Good news for many of us.
Definitely. Then there's resveratrol. That's the compound famous from grapes and red wine. It's also known to inhibit MTOR.
Resveratrol, right.
Curcumin, the active component in turmeric, that's another one. But important note,
Yeah.
curcumin isn't absorbed well on its own. You generally need to consume it with some fat, like in milk or oil, and black pepper, which contains piperine, to boost its bioavailability.
Good tip. Turmeric with fat and pepper.
And finally, EGCG. That's a polyphenol found abundantly in green tea, especially Japanese varieties like sencha or matcha. It's got a lot of research behind it for various health benefits, potentially including autophagy induction.
So spermidine, caffeine, resveratrol, curcumin, EGCG. Quite a list from diet.
Uh-huh.
What about other lifestyle things besides fasting and food?
Exercise is a big one. Physical activity is a known inducer of autophagy.
Makes sense. Stressing the system in a good way.
Exactly. Also, exposure to heat, like using a sauna. This causes heat shock proteins and can trigger autophagy.
Sauna, okay. Interesting. Anything else?
The paper even mentions electrical stimulation. It speculates, and this is more speculative, about a possible link to stimulating the vagus nerve, which might, in turn, activate autophagy. Needs more research for sure.
Electrical stimulation.
Okay,
That's different. So fasting, diet, exercise, heat, maybe even electricity, lots of ways to potentially boost general cleanup.
That covers the main autophagy inducers mentioned, yeah.
All right, so we've talked about directly hitting the spike protein and this general cleanup, autophagy. Now let's zone in on the mitochondria cleanup mitophagy.
Right. Mitophagy, as we said, it's that selective autophagy specifically targeting damaged mitochondria.
Why is that so important? Just getting rid of the broken ones?
Pretty much. Damaged mitochondria aren't just inefficient energy producers. They can actually leak damaging molecules, like reactive oxygen species, into the cell. So clearing them out is crucial for cellular health.
Okay, so it prevents further damage. How does the cell know which ones are damaged?
It involves specific signaling pathways.
Hmm.
Often, damaged mitochondria lose their membrane potential, that electrical charge we mentioned. This can trigger proteins like PINK1 and parkin to essentially tag the damaged mitochondrion, often with ubiquitin.
That ubiquitin tag again.
Exactly. That tag signals, okay, this one needs to go. And the autophagy machinery engulfs it for disposal via the lysosome.
So while autophagy is the general janitor, mitophagy is specifically taking out the faulty power generators.
Good way to put it.
And are there specific compounds that can help encourage mitophagy, help the cell identify and remove those damaged ones?
Yes, the paper highlights several natural compounds thought to induce mitophagy. Resveratrol pops up again
The
here.
versatile stuff.
It seems so. Also, physatin, which is found in strawberries and other fruits, nicotinamide mononucleotide, or NMN, a precursor to NAD plus AC, which is vital for energy metabolism.
NMN, yeah, heard of that too.
Melatonin is another one. We often think of it for sleep, but it has powerful antioxidant and apparently mitophagy-inducing roles in the cell.
Melatonin, interesting.
And methylene blue is also listed here. It's a synthetic compound with a long history in medicine.
Okay, resveratrol, physatin, NMN, melatonin, methylene blue. It's interesting you mentioned some of these are also being looked at for long COVID.
That's right. There seems to be quite a bit of overlap between compounds studied for promoting mitochondrial health and mitophagy, and those being explored as potential supports for long COVID recovery. It suggests mitochondrial dysfunction might be a key factor there.
It really seems to be pointing towards mitochondria being central. Okay, so we can help clean out the bad ones. What about making new healthy ones? That was the next strategy, right? Mitogenesis.
Yes, exactly. Mitogenesis, the creation of new, healthy mitochondria. This is vital for replacing older damaged ones and boosting the cell's energy capacity.
So how do we encourage that? Does nattokinase come back here?
Actually, the paper does list nattokinase again here, suggesting it might have this dual role helping clear out spike protein and promoting new mitochondria.
Okay.
Also listed prominently is PQQ. Its full name is pyrroloquinoline quinone.
PQQ. Okay.
And interestingly, remember natto, the food source of nattokinase.
Yeah.
Natto is also considered one of the richest food sources of PQQ,
Whoa. So eating natto might give you the enzyme in the PQQ.
potentially hitting mitogenesis from two angles mentioned in the paper. Yeah. Natto seems like a bit of a powerhouse in this context.
Seriously. Cellular health superhero, like you said. What else encourages new mitochondria growth?
Cold exposure is a well-known stimulus for mitogenesis. Think cold showers, ice baths.
Okay, cold.
Endurance exercise is another powerful one, though again the paper rightly adds a note of caution. Consider your current health status. If you suspect you have ongoing issues, maybe from spike protein exposure, you don't want to overdo strenuous exercise.
Good point. Listen to your body. How does exercise trigger it?
Several ways, but one key mechanism is boosting the production of nitric oxide, or NO. And nitric oxide itself is a signal that promotes mitogenesis.
Ah, nitric oxide.
And melatonin makes another appearance here, too. It seems to support mitogenesis as well.
Melatonin really does seem multifaceted.
It appears so, supporting mitophagy and mitogenesis.
Okay, so to recap the creation side, possibly nattokinase, definitely PQQ found in natto, cold exposure, sensible endurance exercise, boosting nitric oxide, and melatonin.
That covers the main points for mitogenesis from the paper,
All
yes.
right, so we've covered getting rid of spike protein, general cleanup autophagy, specific mitochondrial cleanup mitophagy, and making new mitochondria, mitogenesis. What's the last piece? Supporting the ones we already have.
Exactly. Supporting the proper function of your existing healthy mitochondria, making sure they run efficiently and are protected.
What helps with that?
Several key nutrients and factors are mentioned. Vitamin C is a big one. As we've maybe touched on in previous deep dives, vitamin C can actually get inside mitochondria and act as an antioxidant right there, protecting them from stress and damage.
Vitamin C. Okay. Simple but important.
Then there's N-acetylcysteine, or NAC.
NAC.
NAC is a precursor to glutathione, which is arguably the body's master antioxidant. So NAC helps boost glutathione levels, providing antioxidant defense for mitochondria.
Okay, but wasn't there a caution with NAC? Something we discussed before.
Ah, yes. Good memory.
Yeah.
In our series looking into turbo cancers, there was discussion around a potential, and it's stress potential, link in some context between high glutathione levels, which NAC boosts, and high IgG4 levels, sometimes seen after mRNA vaccination. It's complex and not fully understood, but it's a relevant caution to mention when discussing NAC, especially in this overall context. Something to be aware of and maybe discuss with a healthcare provider.
Okay, duly noted. What else supports mitochondrial function?
Magnesium. Absolutely crucial. It helps maintain the proper electrical charge, the membrane potential, across the mitochondrial membrane. This charge is essential for the process of making ATP, the energy currency.
So, like keeping the battery charged correctly?
That's a perfect analogy. Magnesium is key for that mitochondrial battery.
Got it. Magnesium.
Methylene blue comes up again here, too. Besides inducing mitosophagy, it can also act as an electron cycler within the electron transport chain inside mitochondria. Basically, it can help electrons move along the chain more efficiently, potentially boosting ATP production, especially if parts of the chain are impaired.
Methylene blue again. But didn't that have warnings?
Yes, very important warnings. It's contraindicated, meaning should not be used during pregnancy, and critically, it should not be taken with SSRI antidepressant medications.
Why not with SSRIs?
Because of the risk of serotonin syndrome. It's a potentially dangerous, even deadly condition caused by excessive serotonin activity. Methylene blue can inhibit an enzyme that breaks down serotonin, so combining it with SSRIs is a serious risk.
Okay, that's a crucial warning. Pregnancy and SSRIs, no methylene blue. Understood. Anything else for supporting function?
Lastly, the paper mentions light therapy, specifically UV light from the sun for vitamin D production, which is important for overall health, including potentially mitochondrial function.
Sunlight for vitamin D.
And also infrared light, particularly near infrared, which is also part of sunlight. This type of light seems to directly interact with components in the mitochondria, like cytochrome stick oxidase, to help maintain healthy function.
Infrared light from the sun.
Yes. And interestingly, there's epidemiological evidence linking avoiding sunlight exposure with higher rates of premature mortality from various causes. It suggests getting sensible sun exposure might be more important than just vitamin D. The infrared component seems beneficial too.
Wow. So vitamin C, NAC with caution, magnesium, methylene blue with serious warnings, and light both UV and infrared from sunlight.
That's the list for supporting mitochondrial function from this paper.
That's a really comprehensive overview, drawing from that figure in the paper. It connects a lot of dots between spike protein concerns and fundamental cellular health strategies. What were the main takeaways the authors emphasized at the end?
They really seem to highlight the potential of autophagy, seeing it as a, potentially valuable tool for health, maybe even something that could be used more clinically in the future.
Using the body's own cleanup crew more strategically.
Kind of, yeah. They also pointed out quite rightly that fasting isn't some new fad. It has a really long history across many cultures, used for spiritual reasons, health reasons, for centuries.
True, it's ancient wisdom in a way.
And we are actually seeing it creep into modern Western medicine now, aren't we? Like, fasting before surgery is standard practice, partly because it's thought to reduce complications.
Right, that's a good point. It's already used clinically in some ways.
So it's about maybe understanding the deeper mechanisms now.
Okay, so as we wrap up this deep dive looking at protecting mitochondria from potential spike protein issues, What's the sort of final thought you'd want to leave listeners mulling over?
I think ultimately understanding these basic cellular processes, autophagy, mitophagy, mitogenesis, and realizing how much our lifestyle choices, like diet, fasting, exercise, even light exposure, can influence them. Well, it gives you a sense of empowerment, doesn't it?
Yeah, it feels less like we're just passive victims of circumstance.
Exactly. It encourages you to think proactively. How can you support your body's incredible innate ability to clean up, repair, and regenerate? How can you build resilience at a cellular level, even when facing, modern environmental stressors like novel proteins?
So it's not just about avoiding bad things, but actively cultivating good cellular health.
Precisely. Thinking about long-term well-being and resilience, starting from the inside out.